synthetic biology offers a way to engineer cells to perform novel functions such as glowing with fluorescent light when they detect a certain chemical. Usually, this is done by altering cells so they express genes that can be triggered by a certain input. However, there is often a long lag time between an event such as detecting a molecule and the resulting output, because of the time required for cells to transcribe and translate the necessary genes. MIT synthetic biologists have now developed an alternative approach to designing such circuits, which relies exclusively on fast reversible protein-protein interactions. This means that there is no waiting for genes to be transcribed or translated into proteins, so circuits can be turned on much faster, within seconds. This kind of circuit could be useful for creating environmental sensors or diagnostics that could reveal disease states or imminent events such as a heart attack. Inside living cells, protein-protein interactions are essential steps in many signaling pathways, including those involved in immune cell activation and responses to hormones or other signals. Many of these interactions involve one protein activating or deactivating another by adding or removing chemical groups called phosphates. In this study, the researchers used yeast cells to host their circuit and created a network of 14 proteins from species including yeast, bacteria, plants, and humans. The researchers modified these proteins so they could regulate each other in the network to yield a signal in response to a particular event. Their network, the first synthetic circuit to consist solely of phosphorylation, dephosphorylation protein-protein interactions, is designed as a toggle switch, a circuit that can quickly and reversibly switch between two stable states, allowing it to remember a specific event, such as exposure to a certain chemical. In this case, the target is sorbitol, a sugar alcohol found in many fruits. Once sorbitol is detected, the cell stores a memory of the exposure in the form of a fluorescent protein localized in the nucleus. This memory is also passed on to future cell generations. The circuit can also be reset by exposing it to a different molecule, in this case a chemical called isopentenyl adenine. These networks can also be programmed to perform other functions in response to an input. To demonstrate this, the researchers also designed a circuit that shuts down cells' ability to divide after sorbitol is detected. By using large arrays of these cells, the researchers can create ultra-sensitive sensors that respond to concentrations of the target molecule as low as parts per billion. And because of the fast protein-protein interactions, the signal can be triggered in as little as 1 second. With traditional synthetic circuits, it could take hours or even days to see the output. The researchers now hope to use their protein-based circuits to develop sensors that could be used to detect environmental pollutants. Another potential application is deploying custom protein networks within mammalian cells that could act as diagnostic sensors within the human body to detect abnormal hormone or blood sugar levels. In the longer term, the researcher envisions designing circuits that could be programmed into human cells to report drug overdoses or an imminent heart attack.